Coming on to our topic, mapping by interrupted conjugation. In mapping by interrupted conjugation, the HFR and the F- cells are mixed together and they are allowed to conjugate. And at fixed intervals of time, the F- cells are sampled to determine which all donor alleles have entered into the recipient cell. Now this sampling is accomplished by separating the joined cells by using a kitchen blender, resulting in interrupted conjugation. And after separation, the HFR cells are selectively killed. How are they selectively killed? We have already discussed in the previous section. We have to consider an antibiotic resistance property or an antibiotic resistance gene for this function. For example, if we consider streptomycin resistance in F- cells and streptomycin sensitivity for HFR cell, and after separation, if we add streptomycin to that medium, HFR cells will be selectively killed and F- cells will be intact. So the HFR cells are selectively killed and the remaining F- cells are tested to see which all donor alleles have entered into the recipient cells and stably recombined with the endogenome, that is the chromosomal genome. Okay, so if a donor allele A plus enters the recipient at 5 minutes, if we interrupt the conjugation at 5 minutes and check the F- cells for the donor alleles, we see that A plus has entered the recipient cell. And similarly, after 8 minutes, when we check the F- cells, we see that B plus allele has also entered the cell. So this means that these two genes, that is A plus and B plus, are 3 minutes apart on the chromosome. And here the map units or the distance units are in minutes. So let's study about mapping by interrupted mating with the typical cross. So in this cross, the genes that we have taken under consideration are AZI, which is the resistance or sensitivity to sodium azide, GAL, the ability or inability to utilize galactose as an energy source, LAC, the ability or inability to utilize lactose as an energy source, and TON, which is the resistance or sensitivity to bacteriophage T1. And these are the genes that we have taken under consideration for studying interrupted mating technique. So the streptomycin sensitivity allele and resistance allele, that is the basis for selectively killing HFR cells that we have already discussed in the previous slide. So how is this selectively killing accomplished? By adding streptomycin to the mixture of cells after interrupting the conjugation. So when we add streptomycin, the streptomycin sensitivity allele in HFR cause the degradation or killing of HFR cells. However, the streptomycin resistance allele in recipient or the F- cells keeps that cell intact. And it will be advantageous if that contrast-selecting allele, in the previous case that we have discussed, it is streptomycin resistance or streptomycin sensitivity, that should be very close to the integrated F factor, or if it enters close to last, because then it will only rarely enter the F- cells. So if it is so close to the integrated F factor, it will be advantageous in mapping by interrupted mating. And for that, the position of the contrast selected gene must be already established. We should know the position of the contrast selecting allele or the gene. So here in this particular cross that we have taken as an example, we have considered these genes: streptomycin sensitivity, streptomycin resistance in HFR and F- cells respectively. Then AZI resistance, sensitivity, GAL plus and GAL minus, where the GAL gene is present, GAL allele is absent. Then LAC plus, LAC minus, TON R and TON S. So in this particular cross, after allowing the cells to undergo conjugation and then interrupting the conjugation at different time intervals, it was detected at 8 minutes that AZ gene entered the recipient, and followed by TON or LAG plus and GAL plus in that order. So we got the gene order as well as the distance between these genes in minutes. And this is the order in which the genes are located on the chromosome. We have selected F- cells which are streptomycin resistant and carry these genes, but we have not taken TON here. Anyway, it carries as a gene LAG and GAL, and it is allowed to conjugate with an HFR cell which is streptomycin sensitive and carry these wild type of alleles. Now, when the conjugation starts, the DNA is nicked at the origin and the DNA strand transfer starts. I hope you remember the conjugation process. And once the F instructs the gene transfer, we start the mating clock. And if we interrupt this mating at 4 minutes and add streptomycin and test for any recombinants among the F- survivors, none are found. Now, if we wait for 8 minutes to interrupt the mating, we find that the azide resistance allele has been transferred. And after 16 minutes of mating, our results show that the donor lactose gene has also been transferred. And after 24 minutes, if we wait for 24 minutes of mating, our results show that the donor galactose gene has been transferred as well. So the recombinant analysis revealed the order of donor genes behind the F origin, that is AZ, LAC, GAL. We discussed about gene mapping in bacteria by interrupted mating. Now let's see the F factors which carry the bacterial genes. We have already learned or discussed about this. That is, sometimes the integrated F plasmid or the F factor moves away from the integrated state back to the cytoplasm, and sometimes while doing so, it might carry some of the bacterial chromosomal genes along with it. Now that F factor is no longer a normal F factor, it is now called an F prime. And F prime is ready to transfer that bacterial gene that it carries through conjugation in an infectious manner to other cells, to the recipient cells. So if an F prime plasmid is transferred through conjugation to an F minus recipient, the recipients are made into stable merozygotes which carry a complete bacterial genome, that is its own genome, plus a donor fragment, that is from the donor a portion of its gene on the autonomously replicating plasmid, that is on the F plasmid. So a stable merozygote is formed when F prime plasmid is transferred to an F minus recipient from an HFR, a previously HFR cell. And that merozygote, it is a merozygote because it contains its own bacterial chromosome and a donor fragment on F plasmid. Now this process of creating a merozygote by an F prime element is called as sexduction or F prime duction. And stable partial diploids are very useful in bacterial genetics because they can be used to study the dominance, to determine which allele is dominant. So it is very important for genetic studies in bacterial genetics. For example, suppose the donor is LAC plus, LAC positive, and that is used to create an F prime LAC plus plasmid. That is, while moving away from the integrated state, the F factor or the F plasmid carries that LAC gene along with it, or LAC allele along with it. And during conjugation, this plasmid is transferred to the F minus recipient, which usually carries the allele LAC minus. So it is LAC negative. And this partial diploid is heterozygous. That is, when this F prime LAC plus enters into the F minus cell, now it contains a diploid condition in which it is heterozygous. That is, lac plus is there from the plasmid and lac minus its own chromosomal genome. And these cells can be used to determine which allele is dominant. So in this case, this partial diploid condition can be used to determine which allele is dominant. And here in our studies, it has been shown that lac plus turns out to be dominant. This is a diagrammatic representation of the above mentioned plasmid which carries a bacterial genome, that is F prime. And here you can see the integrated F factor, which is the HFR chromosome. And then it moves away from that integrated state back into the cytoplasm. So while doing so, it carries some of the, and here lac plus allele or some of the genes of the bacteria. So it carries this lac plus gene along with it. And while conjugation, it gets transferred or it gets copied into the F minus cell, which is usually lac negative. And now it contains a lac positive and a lac negative gene or alleles. So it is heterozygous. And this is a partially diploid cell. So it is a merozygote. And because of this heterozygous condition, this can be used to determine which one is dominant. And as I told you, studies have shown that lac plus gene or lac plus allele is dominant. So that's all what I wanted to discuss in this presentation. I hope this will be very useful for you. Thank you.